I'll be speaking about genetics, and genetics is very easy, as you will see, because most of what we encounter in nephrology is monogenic, so it's Mendelian, it's recessive or it's dominant. And increasingly, many of the diseases that are monogenic will be seen by adult nephrologists because these patients survive the adolescent age now. Here's my disclosure slide. And let's start with this patient. This 3-year-old boy developed periorbital edema and generalized edema as well as ascites. And that was due to nephrotic syndrome, which turned out to be steroid-resistant. His renal biopsy showed focal segmental glomerular sclerosis, and you can see the fibrous strands here in this trichrome Masson stain. And as unfortunately it is the case with FSGS, within a couple of years, he had to enter the chronic dialysis and transplantation program. So if we ask ourselves, why does he have to go through all this, and what is the cause of disease, we are in the exceptional situation here that we can actually find the cause. And it's shown in this slide. He carries a homozygous point mutation where this triplet codon GCG, which encodes the amino acid alanine, is exchanged for a thymine here, GTG, which encodes the amino acid valine. Thereby the structure of the protein, in other words, the amino acid sequence, is altered, and thereby structure and function of this protein called Podocin, where the gene is called NPHS2, is defective. And a situation like this, in my mind, is one of the strongest cause-effect relationships that we can see in clinical medicine, in that a single mutation in one base pair of the about 3.3 billion base pairs that we have in our genome is sufficient in itself to cause disease. However, as you know, this is just characteristic for thousands of diseases that we call monogenic, or that we call single gene disorders or Mendelian disorders. And they're defined by the fact that in a given patient, the disease is caused, and I emphasize caused, by a mutation in one gene only, as in this child, out of the about 20,000 genes that we have in our genome. This mutated gene can be a recessive disease gene, where recessive means as we have 2 copies in most of these 20,000 genes, one from mom and one from dad, both copies have to be mutated for disease to occur. So the parents will be healthy heterozygous carriers. They will both each contribute a mutation only from one of their chromosomes, and no one in the ancestry will ever have had disease, because at worst they're also healthy heterozygous carriers for this recessive gene. And this, for instance, is the case in ARPKD, autosomal recessive polycystic kidney disease. In contrast, in a dominant disease gene, a mutation in one of the 2 copies is sufficient to cause disease. Therefore, one of the parents will have the disease most likely, and the disease will have traveled throughout a pedigree for all the heterozygous individuals. And this, for instance, is the case in autosomal dominant polycystic kidney disease. The term monogenic or single gene disorder or Mendelian disorder does not exclude, however, that in different patients, different genes may cause a very similar indistinguishable disease. And this is called genetic locus heterogeneity, where locus just refers to the fact somewhere in the genome there is this gene, and you have multiple genes that can cause a similar disease. For instance, FSGS can be caused by Podocin mutations or nephrin mutations. So right now, there are about 42 recessive monogenic causes of focal segmental glomerular sclerosis or steroid-resistant nephrotic syndrome, and 8 dominant genes, and a novel monogenic cause of steroid-resistant nephrotic syndrome is currently detected or discovered about every 2 months. So this number will keep rising for a while. So why is it, what is special about monogenic diseases? Let us look at the flow of genetic information as it was formulated in the so-called, central dogma of molecular biology by Francis Crick. Because there's a virtually deterministic flow from the gene to the phenotype, that's why I put red arrows here, where a gene is expressed as a messenger RNA, it leaves the nucleus, it's translated into a protein in the cytosol, And I don't know my Greek, but I heard proteo means I'm the boss, I'm the most important. So proteins are called that because they're very important for structure and function of the organism. So the proteins together determine a certain phenotype. In a monogenic disease, the mutation itself, be it recessive or dominant, is identical with the primary cause of disease or the etiology of disease. So the mutation will be expressed as an altered messenger RNA, which may lead to a truncated protein or a structurally altered protein, as in our patient, and thereby cause disease. Now, why is it important to know the mutation by mutation analysis? With the gene in hand, as it represents the primary cause of disease, we can then offer to the patient and sometimes the family unequivocal molecular genetic diagnostics, and often interrupt a diagnostic odyssey that these patients undergo. With the gene in hand, we can now study this black box, which we call pathogenesis or disease mechanisms, where there's very little known in nephrology so far. Sometimes we discover some new physiological mechanisms in this process. And then often we are able to draw genotype-phenotype correlations, where, for instance, mild mutations may cause late onset of disease, more severe mutations may cause early onset of disease, or sometimes more severe mutations may include extrarenal manifestations in some of these genes. And increasingly so, we hope that by knowing the specific mutation, we are able to either select a more specific therapy or even develop a more specific therapy in way of precision medicine. So I would like to parallelize one more time monogenic mutations versus complex genetic disorders. In monogenicity, when we look at a genetic variant, that means a nucleotide that's different from a control group, we usually call it a polymorphism. If we have functional evidence or genetic evidence that it causes the disease, then we can call it a mutation. So the term mutation implies this is causative. Whereas in complex disorders, as you will see, only multiple genes together can be a risk factor for a disease, and this is not causal, this is called an association. So on the left column you see monogenic disorders again, where in a recessive case this child would be homozygous for a mutation, has a healthy heterozygous sibling, healthy heterozygous parents, and usually recessive disease genes show full penetrance, which means everyone who has the two alleles mutated will develop disease by a certain age. Whereas in complex disorders, only alterations in multiple genes together explain a genetic risk in a population, so that the inheritance in a pedigree will not logically look like a dominant disease or like a recessive disease. If we one more time look at disease causation by genes rather than by the environment, or we look at this gradient from nature to nurture, from red to blue, and let's start at the blue end here, then you could make a case that trauma is external pathogenesis. However, there could even be a genetic predisposition for someone to suffer from who is accident prone, so there could be a little bit of a genetic component already. Toxins are really external. However, there are genetic predispositions for toxins, such as in G6PD deficiency. Infectious diseases are external, but again, there are genetic predispositions. Degenerative diseases are a mixture of external degenerate influences and, again, genetic predisposition. Developmental disorders are already shifting more towards genetic influences. Most metabolic disorders are just inborn errors of metabolism that are almost monogenic. In polygenicity, we've looked at that. You need multiple genes and only a minor external influence. And then in the true monogenic diseases, dominant or recessive, the determinism in dominant is not as strong because you can have what's called incomplete penetrance, but in recessive diseases, there's mostly full penetrance by a certain age. Now, here we go. I want to quantitate this a little bit. Now let's look at our lifespan from birth to 100 years of age. And now let's look at the cumulative likelihood that the disease will develop in multiple individuals. And let us look at individuals with mutations in an autosomal recessive gene. For instance, the Podocin gene that can cause FSGS. If there is a homozygous protein truncating mutation, this really alters the protein, and it may be that this S-shaped curve shows you that about age 12, most of these individuals will have developed disease. That's called full penetrance. But what we see in many of these Mendelian disorders is that there's allelism. That means the specific mutation determines what the clinical phenotype is. And to keep things easy, I'm only looking at homozygous recessive mutations. So here's a truncating mutation on both parental alleles, and here's a missense mutation on both parental alleles. So it's a pure representation of this mutation. And a milder mutation often shifts this curve of manifestation to the right with later onset and milder course. Dominant diseases often have a tendency to manifest in adult life. And there is this feature in dominant but not recessive that there is what's called incomplete penetrance. In other words, a patient does have this mutation that the other individuals who are affected in the pedigree have, but this patient never develops disease, even if he or she becomes to be 100 years old. There's many ill-understood mechanisms that underlie incomplete penetrance. And then there can be, again, allelism. That means there can be a mild allele in the same gene, and these individuals may manifest later and may not even live to see their disease. Now, very rarely, and I don't want to confuse you, usually it's very clear-cut. A mutation is recessive or behaves in a recessive way. You need 2 for disease to occur, or a mutation is dominant. You only need 1 for it to occur. Very rarely genes, for instance, nephrolithiasis genes, can have both forms of Mendelian inheritance, and that depends on the specific mutation. Certain mutations act in a recessive way, other mutations act in a dominant way. But that's very rare, and don't get confused. So now, Mother Nature can make things worse. Environment influences on the basis of these strong genetic influences can shift the severity over to the left, an earlier occurrence. And we as physicians hope that prophylaxis and treatment can shift these curves over to the right to mitigate these diseases. Sorry, here we go. So here's a board's question. Maybe. So the term monogenic disease, A, is synonymous with single gene disease and Mendelian disorder. B, describes autosomal recessive, autosomal dominant, and X-linked disorders. C, describes a genetic association. D, means that only 1 gene in an individual's genome is the cause of disease. And we've emphasized that the term association is usually used for polygenic disorders. So this combination, A, B, and D, looks good to me. Okay, let's look at causes of chronic kidney disease, since I'm a pediatric nephrologist, in individuals who manifest with CKD or chronic kidney disease before age 25. And increasingly, luckily, these individuals survive into adulthood, and you as adult nephrologists see many of the diseases that about 20 years ago you would not have seen in adult nephrology. This is a study, NAPRTCS, North American Pediatric Renal Transplant Cooperative Studies, where it was looked at 9,000 individuals and the cause of chronic kidney disease in this age group. And as you know, most of these individuals develop chronic kidney disease because of congenital anomalies of the kidneys and urinary tract. In other words, renal agenesis, vesicular-ureteral reflux, hydronephrosis, ureteropelvic junction obstruction, and so on. 15% have what our initial patient has, steroid-resistant nephrotic syndrome. 14% have a sort of chronic glomerulonephritis. 6% have some sort of cystic kidney disease, ARPKD, ADPKD, nephronophthisis, and what you've just heard over lunch, medullary cystic kidney disease with UMAT mutations, for instance. And I also put nephrolithiasis here because recently that has some genetic relevance. And only in the last, say, 8 to 10 years, it was realized that for many of these individuals, there are monogenic genes that cause them. Right now, as it stands, there is already 46 genes for congenital anomalies of the kidneys and the urinary tract. 2 years ago, this was 12 genes. So this is very rapidly growing, and there's going to be hundreds. Steroid-resistant nephrotic syndrome, now there's 50. 2 years ago, there were 30. 8 years ago, there were 5. In cystic kidney diseases, there is 95. When I was a fellow, there were 0. In nephrolithiasis, there's at least 30 genes that cause recessive or dominant forms of these diseases. And only in the last 2 years, it was realized that if we look in all these genes, over 100 of them, for instance, by whole exome sequencing, because you know the exons are the business end of the genome, it's only 1% of the genome, but this is where the proteins are encoded. And luckily, 85% of all monogenic disease mutations are in exons or in the splice sites that are right next to them. So you have, in theory, an 85% chance of finding a causative mutation in a monogenic disease by just running whole exome sequencing, which right now kind of has a base price of $700, but most companies charge $3,500. So only in the last 2 years, it was realized if you run the exome, you can find the cause of disease by mutations in any of these genes in 17% of the individuals with congenital anomalies of the kidneys and urinary tract, or COQ6, in 30% of children with steroid-resistant nephrotic syndrome and young adults, in 70% of individuals with a cyst in the kidney, or cysts in the cystic kidney disease, I should say. And very surprisingly, last year we saw that in children you can find a monogenic cause of nephrolithiasis in 20% and in adults in 11%. So when we asked in a worldwide cohort, in what percentage can you find a monogenic causative mutation in steroid-resistant nephrotic syndrome that the initial patient had, and to make a long story short, in 1,700 worldwide families, we found a causative mutation in 30% of these individuals, being careful to only call convincing mutations that were truncating or highly conserved amino acid exchanges. And the most prominent gene is Podocin, next one is nephrin, Wilms tumor 1 is 5%, PLCE1 is 2%, and then there is dozens of more rare genes, and as I said, there will be one new gene every 2 months right now, and this number will slowly increase. However, this fraction, which has been confirmed in European studies, which is surprisingly high, has to be taken with a grain of salt because it's age dependent. So in infants who have steroid-resistant nephrotic syndrome, the likelihood to find a monogenic cause is even 60%. Then in toddlers, it goes down to 20%, and in young adults, it's around 10 to 15%. There's also a genetic geographic distribution difference. I'm showing you here Europe and the Middle East. And you know, in recessive diseases, cousin marriages mean a risk factor for recessive diseases, because if you think of the fact that mom and dad are related, if an ancestor to mom and dad has a heterozygous mutation in a recessive kidney disease gene, and actually we all have 10 very bad recessive mutations in 10 genes, but we're heterozygous for them, so we don't know it and it doesn't do us any harm. But most people have these 10, so if the ancestor has a heterozygous mutation in the Podocin gene, there's a high risk that it'll travel through the dad's side to this child and the mom's side to this child, and the child will be homozygous. It will have both alleles mutated and thereby develop the disease. Everybody will be a healthy heterozygous carrier, and no one in the ancestry will have had disease because they're all heterozygous, but the child is at a higher risk, actually a 1 in 64 risk that it'll come together if they're first-degree cousins. So when you do the whole exome, you look in all these nephrosis genes, then there is a clear correlation between the fraction in these cohorts of consanguinity, which goes here on the x-axis from 0 to 80%, and the likelihood to find a causative mutation in one of these 30 genes, from 15% to 45%. And again, the most prominent genes were Podocin, Nephron, WT1, and then the mixture of others shown here in blue. In the US, we have data that is in the range of 10 to 15%. Okay. Whoops. Okay. Now, if there's this high fraction of renal disorders in which we can now find a monogenic cause of disease, why did we not realize that earlier? I think there's two major reasons. One is, just 5 years ago, we couldn't have paid for it because exomes were very expensive, so it was not technically feasible or it was not monetarily feasible. The other reason is, when we think of genetic causation, we immediately think familial occurrence of disease. So we do a family history and there's nothing in the ancestry. But familial occurrence should actually not be the case in recessive diseases. Because if you look at it, in this family with 4 kids, even if dad and mom are both heterozygous carriers, say of a Podocin mutation, they will stay healthy for the rest of their lives. Because, as I said, we all have 10 very bad heterozygous mutations in recessive genes will never develop disease because it's heterozygous. The other allele completely compensates. That's the definition of a recessive disease. Now, according to Gregory Mendel, there's random assortment of chromosomes. So statistically, of the 4 children, 2 of them will inherit the mutated chromosome from the dad, 2 will inherit the healthy one. Same for mom, 2 will inherit the mutated from the mom, 2 will inherit the healthy one. So that statistically, only 1 child out of 4 will have both alleles mutated, thereby develop FSGS. There will be 2 healthy heterozygous carrier children, and there will be 1 homozygous wild-type child that doesn't even carry the disease. So statistically, you would have to have 8 children for 2 of them to have disease. Since publications want to go for extreme cases, the families who have multiple children with the disease get published, and that's what skews our mind that we expect familial inheritance in recessive diseases. But if you have 10 children or young adults in your clinic with FSGS, according to the statistics of up to 30% monogenic inheritance, and you take the family history in each one of them, and it's completely negative, that doesn't exclude at all that their disease could be caused by a recessive gene. Let's get back to steroid-resistant nephrotic syndrome. If you're as old as me and you went to the American Society of Nephrology meeting for the last 28 years, then for the first 20 years or so, there were hundreds of abstracts that tried to incriminate this blue cell, the mesangial cell, in the pathogenesis of steroid-resistant nephrotic syndrome. So, you know, here's the capillary with the fenestrated endothelia in yellow. There's the mesangial cells that holds the tuft together. There's the basement membrane in green. And then there's the epithelial cell layer, the so-called podocytes. And the podocytes are neuron-like cells that are shown here, and one is shown here on the right. It looks like an octopus. It hugs the glomerular capillary with its primary processes that split off into secondary processes, and then you cannot see that, but again, they split off into tertiary processes, which, as you know, are the foot processes that interdigitate to form the glomerular filter, to retain the protein in the bloodstream. And in nephrotic syndrome, this filter is disintegrated. So if we actually, it's always neighboring podocytes that form this filter, so the yellow one will hug with a blue podocyte or a red one. If we cut through the glomerular slit membrane, as shown in the next slide, and you see the podocyte here, this is my beautiful rendition of a podocyte with a very huge foot process and the neighboring foot process over here sitting on the glomerular basement membrane, then through high throughput genetic sequencing, what has occurred is that these 50 genes were found that you see here in red and blue bubbles. So the bubbles denote proteins. For the red ones that are encoded by genes that, if mutated, cause recessive steroid-resistant nephrotic syndrome. The blue ones are proteins encoded by genes that, if mutated, cause dominant nephrotic syndrome. And what has happened fascinatingly in the last 5 years is that all the genes that were found by genetic mapping and whole exome sequencing point to proteins that are actually part of protein clusters. So I put them in these black frames here. And when you look at what the cluster does, you can assign a function. So by finding all these monogenic causes of disease, we can reconstruct what a podocyte needs to be a happy podocyte. So nephrotic syndrome, in a way, is a podocytopathy. It's disease of an unhappy podocyte. So these functions are now related to integrin laminin receptors, where the podocyte is tethered to the glomerular base membrane, actin binding proteins, Rho-RAC regulation, and so on. Some transcription factors play a role, but I would like to briefly give you an example on enzymes that are part of the biosynthesis of CoQ10, which is kind of a food supplement and kind of a famous food supplement with no side effects. In children where recessive mutations in genes, these genes were found, some of them responded to treatment by replenishing just CoQ10, because when finding these genes, we were able to show that the cells lack CoQ10. For instance, here's this 6-year-old girl who had a mutation in this enzyme COQ6, which is important for CoQ10 synthesis. And she was given CoQ10. Then the parents, while she was in remission from a proteinuria, then the parents forgot to give the drug. And when they reintroduced the drug, she had a remission, or at least a partial remission, from her nephrotic syndrome. Now, so rarely though, really rarely, but already, detection of monogenic cause of steroid-resistant nephrotic syndrome can influence your management of the patient. And this hopefully will be increasingly so for the monogenic diseases. Now, we've talked about steroid-resistant nephrotic syndrome and its 50 genes. I would now quickly jump to cystic kidney diseases. And you have heard that cystic kidney diseases have been referred to as so-called ciliopathies, because primary cilia, which are antenna-like structures that virtually every cell in the body has or can have during development, are affected, or at least proteins that localize to cilia are affected. Some of the proteins don't really localize to the cilia, but they localize to their anchors, which is the so-called basal body down here. So what was found out that in these 95 genes that can cause cystic kidney diseases, and here's the cystic kidney, in us or in all these animal models, in mice or fish, that these proteins actually localize to cilia. And again, they cluster and point us to some of these functions. So you have cystic kidney diseases. Some of these are associated with retinal degeneration, some with liver fibrosis, some with diabetes, some with situs inversus, and there are all these difficult names that you'll see: nephronophthisis, Bardet-Biedl syndrome, Senior-Loken syndrome, Joubert syndrome, Meckel syndrome, where these disease names actually usually describe what organ is involved. Senior-Loken syndrome would, for instance, include retinal degeneration and night blindness. So what we can do already is to go from a non-invasive, very non-invasive clinical test, which is a renal ultrasound, to an etiologic diagnosis by running whole exome sequencing. So if you go to a cohort, which we did here, of individuals before age 25 who either have 2 or more cysts in the kidney or increased echogenicity. So this is a very easy entry criterion, a very non-invasive test. Just see, is the kidney as white as the liver, or is it even whiter? Then in 79 individuals, we ran the exome and found in this blue section causative mutations in one of these 95 genes, and thereby gave them an etiologic diagnosis. In the red segment here, we found genes that hadn't been described by then. And then if we take this section of the 63% where we found a cause and split it up, then actually in some of them the diagnosis wasn't right. There were many renal tubulopathies, and I'll show you examples in a second, sneaked in. There was some Alport patients sneaked in which weren't diagnosed. There were ARPKD, which kind of belongs there, and there were some congenital anomalies of the kidneys and urinary tract. And this was all under in patients where the diagnosis wasn't clear, but there was increased echogenicity or 2 or more cysts on an ultrasound. So by running the whole exome sequencing under this inclusion criteria, you often can jump directly to an etiologic diagnosis. I want to give you a few cases. This patient, this is very clear. The kidney is really echogenic. There's loss of corticomedullary differentiation, and you have these cysts. And that, in fact, was a true nephronophthisis gene. It was a recessive mutation in NPHP4. Then we had, I'm not showing you the ultrasound here, but it looked similar. We had a patient with these infiltrates and some cystic dilation of tubules, which looked like nephronophthisis, but he had a mutation in a very rare gene called SLC41A1, and that's a hypomagnesemia gene, which can mimic nephronophthisis. Here you would have said probably as a nephrologist already, this, okay, is increased echogenicity, but it has a pepper and salt pattern, so maybe it's ARPKD, and in fact this was ARPKD in the mutation in the PKHD1 gene. And then in this patient, again, increased echogenicity and a few cysts, but it was hyperoxaluria type 1 with a mutation in AGXT. And we see this increasingly. These patients don't get diagnosed early enough, which has therapeutic consequences. These were siblings, 2 brothers who had echogenic kidneys and cysts. And here you may have said already, wow, with this pyramidal picture here, maybe this is RTA, and it was in fact distal RTA with a mutation in the anion exchanger 1 gene. Okay, whole exome sequencing may help you to reveal a causative diagnosis in a different differential diagnosis, where just by the criterion of cysts or increased echogenicity in individuals who have this before age 25 can lead you to an etiologic diagnosis by whole exome sequencing. Just in summary on the cystic kidney diseases, something similar happens here with the 95 different ones, and this is showing only 40 from the nephronophthisis type diseases. They also cluster now in protein-protein interaction complexes that are part of the machinery of ciliary signaling and centrosome-related signaling. Now, let me just go one ahead. I would like to quickly talk about tubulopathies, just in one slide. Very recently, when we looked into 30 candidate genes, we found that in 15% of 290 individuals with nephrolithiasis, by running the 30 candidate genes, we can identify a monogenic cause of disease. And this was 11% in the adult patients and 21% in the pediatric patients. And we've just confirmed this in the order of 18% in another pediatric cohort. And if you think that, I think 1 in 10 people on this planet have at least 1 stone in their lifetime. So this is really a frequent disorder, and it might help to prophylax recurrent bouts of nephrolithiasis if you can fairly non-invasively, just with 1 blood sample, run the whole exome, or even with a spit sample of 4 milliliters of saliva, which is fairly easy to obtain. Now, in congenital anomalies of the kidneys and urinary tract, or COQ6, I want to give you a quick case. A 7-year-old boy with cystic kidney disease on renal ultrasound, hyperuricemia, and an atrial septal defect. And as you may remember from lunchtime, hyperuricemia, very interesting. Had normal renal function still, and blood pressure was normal, urinalysis was normal, no other extrarenal abnormalities. He had the hyperuricemia, slightly low magnesium. He had been diagnosed with autism spectrum disorder and had, again, a positive family history, as his father, and then on the father's side, paternal uncles, paternal grandmother had a similar condition. So this really sounds dominant, doesn't it? Okay, here's the ultrasound, increased echogenicity, some loss of corticomedullary differentiation, and then in the left kidney even tiny cysts. And here's the pedigree. The affected individuals are in black, so it is really on the paternal side. And I've only talked about recessive and dominant. I've not mentioned X-linked, but X-linked is very easy. Whenever you have male-to-male transmission, it can't be X-linked. Why? Because if a father wants to have a son, he has to give him his Y chromosome, not the X chromosome. Then it's not going to be a son. So this excludes X-linked inheritance. So we would consider this could be medullary cystic kidney disease, which is dominant. There's the type 1 with mucin 1 mutations. There's the type 2 with UMOD mutations, as we have heard this very, very interesting case by David Mount over lunchtime. Or it could be renal cyst and diabetes syndrome, or RCAD, which has HNF1 beta mutations. And I'm showing you this case, which is not fully solved, but just to, as an example. As you heard, uromodulin can cause, mutations can cause medullary cystic kidney disease type 1, type 2, or familial juvenile hyperuricemic nephropathy, or glomerular cystic kidney disease. But in this patient, UMOD sequencing was negative. Also, the alternative diagnosis, HNF1 beta, was negative. However, it's known that most of HNF1 beta mutations are large heterozygous deletions, and you cannot catch them by whole exome sequencing because you will just get point mutations. You cannot dose whole exome sequencing. You will not see that a long stretch over the entire gene is missing on the chromosome from one parent. But in the region of the gene, what we found, there were no heterozygous markers, and I'll show you why. This is a situation, say, in this patient, this is the paternal chromosome on the left and the maternal on the right, and you do all these whole exome sequencing, and you find every position here heterozygous. So in other words, there's a green and a red readout, or there is 100 base pairs and 102 base pairs. So there's heterozygosity from mom and dad. Now, in the region of the gene, there could be everything homozygous, if they're related, and you cannot distinguish that from a deletion in one of the parent's chromosomes. So in other words, if you have a run of what looks like homozygosity, and that's called hemizygosity, then this could either be just homozygosity, or it could be a deletion. What you have to do is you have to run copy number variant analysis, and that's a different test than a whole exome. So you have to ask for CNV analysis to show the presence of this heterozygous deletion, which is most likely the case in this patient. HNF1 beta associated disease, or renal cyst diabetes syndrome, is a multisystem disorder. What is characteristic is the so-called maturity onset diabetes of the young MODY type 5. There can be pancreatic hypoplasia, very rarely genital malformations. There can be elevated liver function tests, and characteristically there's hyperuricemia and often hypomagnesemia, as in this family. Clinical implications in finding the molecular cause could be, for instance, that the patient, the family now knows what is going on clinically and why they have this kidney disease. They may be able to avoid unnecessary diagnostic procedures. And early detection may help with monitoring the hypomagnesemia, uric acid levels, because now we know the cause. It may expectantly help us to avoid that they manifest with ketotic acidosis from MODY diabetes type 5 when they manifest in their 40s. And following transplant, we may be wanting to be careful with pro-diabetic drugs. And if there's a potential living-related donor which hasn't reached the age of manifestation of the disease yet, we should be careful in excluding that he or she is a carrier of the mutation, because then we would only do harm in admitting him as a donor. Now finally, in this list, in these disease groups of steroid-resistant nephrotic syndrome, if we just use the non-invasive entry criteria of persistent proteinuria, this is all before age 25, we have a plus minus 30% chance of finding a causative mutation in one of these 50 genes. If we look in an echogenic kidney before age 25, we already have a 70% likelihood of finding a causative mutation. In congenital anomalies, we have about 17% chance. In stones, between 11 and 20%. So this number of known monogenic causes of kidney diseases will very, very rapidly rise, and many of these causative mutations can already be identified by whole exome sequencing. So today, probably every patient with a kidney disease that may be caused by these single gene mutations should have a chance at having this mutation identified if they so wish. Why? It can now be done, but that's not a good enough reason to do it. But it would also provide the patient with an unequivocal etiologic diagnosis and also the family. It may already rarely have consequences for treatment. It allows us to classify patients for therapeutic trials much better rather than FSGS, really going by etiology. We may understand the pathways better and eventually may be able to develop personalized medicine based on the etiology. Thank you.